An action potential travels from the brain down the spinal cord to a motor neuron. The dendrites on the motor neuron pick up the signal and it is carried through the motor neuron along the axon until it reaches the axon terminal. There, acetylcholine, a neurotransmitter, is released from the axon terminal. It travels across the synaptic cleft and then binds to acetylcholine receptors. The enzyme acetylcholinesterase breaks down any extra neurotransmitters.
Action potential is represented by the orange bead. The clear half tube represents the sarcolemma. The toothpaste box represents the sarcoplasmic reticulum. As the action potential gets carried across the sarcolemma, ATP is needed for the potassium-sodium pump to carry the action potential along. The sarcolemma forms the T-tubule, which carries the action potential into the sarcoplasmic reticulum. Along the T-tubule, there are DHPs. The DHP are receptors that signal the sarcoplasmic reticulum to release calcium. The calcium is represented by the big red bead. For this process, ATP is needed. The calcium ions will connect to the tropamine, which is connected on the tropomyosin on the actin. Once the calcium is connected, it allows the tropomyosin to be reconfigured, opening the binding sites for the myosin head. Once the binding sites are open, the myosin head will be reconfigured into high-energy configuration. ATP is needed for this process. Then the myosin head will connect to the binding site, which is called the cross bridge. ATP breaks down into ADP and phosphate. This causes the myosin head to go from high configuration to low configuration. Because it's still connected, it drags the actin along. This is called the power stroke. In order for the cross bridge to be broken, ATP is needed again. Once the cross bridge is broken, if the contraction is done, the tropomyosin will move back into its original configuration, covering the binding sites once again. Rigor mortis is when the concentration of calcium and ATP are not regulated due to death. Because of this, all the calcium and ATP are released, causing full muscle contractions. Due to the fact that each muscle contracts to its limit, there is not enough ATP to break down the cross bridge.